So what I'd like to do today is to tell you a little bit about my background and the areas of research that I've been looking at in Antarctica for the past 32 years. So when I was a graduate student, minding my own business in the lab one day, I felt somebody tap me on the shoulder, and I turned around. It was my major professor, and he says, "Would you like to go to Antarctica with me? I just got this grant, and there's room for one student." And I said, "Yes." And so I went to the Antarctic for 3 months and came back, and we published a number of papers. And next thing, I was on a postdoc, and I was able to get back to Antarctica. So by the time I became an assistant professor of biology, I had quite a record of working in Antarctica and publishing there and knowing the logistics. And so when I wrote my own proposals with colleagues, it was a very much collaboration. We've been very successful in continuing this program in Antarctica. So there's 3 areas that I'm doing research in. I've gotten very interested in climate change, and I hope you'll come tonight to hear me talk about the impacts of climate change on the Western Antarctic Peninsula, which are very dramatic, and the implications thereof. But today, I'm going to take you into a realm that we've been looking at for many, many years called chemical ecology. And we're going to look at this area of chemical ecology. More recently, I've actually gotten involved with a project where we are taking ships to Antarctica and dragging submarines up the Antarctic slope and onto the Antarctic shelf to photograph king crabs that for the first time in millions and millions of years, we believe, are beginning to climb up towards the waters of Antarctica as the Antarctic circumpolar current is warming. And I'll tell you a little bit more about that tonight. All right, so let's see if I have a... So one of the things I've been doing is writing books for the general public. I'm interested in taking the science of Antarctica and my background in science out to the public and using it as a vehicle to communicate about climate change and ocean acidification and doing science. Science is a funny word now in our society in America. There's a need for scientists to step up and talk to the public about science, and this is something that I've decided is important. This is the book that Dr. Orions mentioned, and then this book came out last week. It's called A Naturalist Goes Fishing, and you don't have to fish to like this book. It's a combination of nature and information about climate change and ocean acidification and overfishing and wonderful adventures around the world fishing. All right, so I've worked in 2 places in Antarctica. I've worked at McMurdo Station for about 12 years back in the 80s and 90s, which is the largest station in the world in Antarctica. There are 1,000 Americans there during the summer, several hundred during the winter. But about 15 years ago, our chemical ecology work moved from McMurdo Station up to Palmer, which I always had heard of was the French Riviera of Antarctic stations. It was the banana belt, they said. Instead of 1,000 people, there were 44, and you were nestled on a beautiful point with a glacier behind you. And you could dive in the waters without having to drill through 10 feet of ice like we were doing. It sounded wonderful. So this is where I'm working now, is up here at Palmer, in large part because there's algae there as well as invertebrates, and we can study those. So how do I get to Palmer? I get on a plane. I fly to Santiago, Chile. From Santiago, I get on another plane and fly another 3 hours to Punta Arenas, Chile, at the very southern tip of Chile, and we get on the National Science Foundation ship, which is not great news. It means it's 4 days to get across the Drake Passage, one of the most notoriously rough bodies of water on our planet. Sometimes it's Lake Drake, sometimes it's 30-foot seas. It depends. I've seen them all. But after 3 or 4 days, you see the little arrow up there is pointing at Anvers Island, and going across the Drake and seeing things like this when you start to get close to the continent is just absolutely spectacular. And then you start to notice the wildlife, you know, penguins everywhere. There's 3 species of penguins. The station where I work is just around the corner. This is what it looks like when I'm coming to the station. And this is taken from Palmer Station looking out. And this is the taxi, we call it the Big Orange Taxi, which listed 30 degrees when it was launched after being built. so they had to pour cement on one side of it to get it back upright. And then it was too low in the water to go up to a dock, so they had to build these big giant sort of ballast tanks on either side of the hull, which are filled with air to bring it back up. And then, of course, it gets terrible gas mileage, you can imagine. All right, so here's home away from home. 44 beds. The main limiting factor is beds. We have about half of those beds filled with scientists and about half of them filled with a doctor and carpenter and a plumber and 2 cooks and people that go out and, survey the glaciers and do science experiments, science techs, I guess you would call them, not so much doing the science themselves, but recording the data. This is where I live in the biology section room. It's called the biolab. There's a dive hut here that we use for diving in the water. The water here is warm. The water is about 1, maybe 2 on a warm day, but it's not that minus 1.8 over at McMurdo, so it feels much warmer. And the people that work here are great. There's all sorts of folks that work around the station. This is actually one of our friends here today knows Chuck Amsler and Maggie Amsler, who are my good friends and colleagues that work with me on chemical ecology. And he is a phycologist, and I work with invertebrates, so it's a nice sort of pairing. And here's one of my research teams down on the ice, Chuck and I and Maggie and others. So, chemical ecology. So when we got into the field of chemical ecology, it was largely terrestrial. There's been a ton of work done on terrestrial plants and their insect herbivores and all sorts of interesting interactions. Not so much in the marine realm. That's a much newer field. So we were interested in looking at some very fundamental questions. One of them published in Science was, there is nothing defended chemically in Antarctica. Why would anything be chemically defended? Because there's not really very many fish, predators, so they wouldn't really drive the evolution of chemistry. If nothing's eating these things, then why should they have chemistry that defends them? And it turns out, it was just wide open. We could go down and take a look. So that was sort of one of the premises. And then if we found something chemically active, we were interested in knowing if it prevented feeding, if it kept things from growing on you. If you're a sponge or a coral or something, this is very important. You have to stay nice and clean. And then maybe there's some chemistry that's used for chemical warfare. Species that use chemistry to overgrow one another, this kind of thing. And then because of a wonderful chemist, it's not easy to find a chemist who's willing to put on a dry suit and go into the ocean. But I found one. His name is Bill Baker. He's at the University of South Florida, and he's been working now with me for 25 years. And Bill's job as a natural products chemist is to kind of figure out what chemicals we're dealing with and look at things from the chemical perspective. So one of the really neat things about the field of chemical ecology, it's interdisciplinary. I don't know anybody who's truly a chemical ecologist in the sense that they can do all the ecology and all the chemistry. It's typically a synergy between a group of people. So when we were at McMurdo, we had to get through 8 feet, 10 feet of ice. Now this was quite easy near the station because you just gave a big box of candy bars to the guys with a big tractor, and he drove out onto the ice and he had a big drill. In 10 minutes he could drill you the hole. You put a little fish hut on it and you've got a dive hole. In the field, not so easy. So we would go out by helicopter and we would camp and we would collect animals from different areas of the sound because there's different species and we wanted to sample a broad diversity. And so we had to take a dynamite guy. His name was Queaton. Queaton had a ponytail. He looked like he just got off a Harley, and he had 100 pounds of dynamite in one hand and a bottle of Jack Daniels in the other. And he knew his stuff. So we would drill the hole, and he would then put the dynamite, and one of my grad students would get to push the plunger, and then we'd have this big hole, and we could dive. So here we are. We're diving in dry suits. This technology is fairly new. 25, 30 years ago it started. It allows you to get into water that's very, very cold, and you can stay warm for about 27 minutes, and then your fingers and toes get numb. So this is me getting ready to go in at McMurdo. And I wish it was dark in this room because these under ice pictures are spectacular, but you're looking up at a ceiling of turquoise blue above your head. The first time I dropped below the ice, I was almost fainted because you can see 500 feet. On a clear day, 1,000 feet underwater. And if you're a diver, you know that 100 feet in the Bahamas, you're going, oh my gosh. So it's unprecedented visibility, and that's a story in itself. Very little plankton most of the year. Here on the bottom, we're collecting invertebrates for our chemical ecology work. Now at Palmer Station, it's a little different because we don't have solid ice. Sometimes there is ice, and we have to dive from the shore and sort of go in off the rocks. It's not very pleasant, but we've done it. More often, we use Zodiacs. We go out in these rubber boats. There are 22 small islands around the station within about 3 miles of the station, and we're allowed to go to all those islands and to dive off the shore in this environment out of Zodiacs. And here we are in our Zodiacs going out for a dive and then getting suited up. And the one thing that we have to worry about when we're diving in Antarctica are leopard seals. So there are no sharks in Antarctica, but there are leopard seals. And leopard seals are about 10 to 12 feet long. They weigh 1,000 pounds, and they're incredibly aggressive. So we do not fool with these guys. If we see a leopard seal, we have, I don't know if I have a picture of it. Well, there's a leopard seal. We have a leopard seal recall device that we drop in the water from the Zodiac and turn it on. It's a siren. So the divers hear the siren and they come right up out of the water, get into the boat, and we go to another spot and dive. We don't stay with a leopard seal because they will come in and circle very close, and they are the boss. Clearly, you know that they're the boss. And there's a leopard seal showing off its teeth for you. Would you dive with that animal? No. And here's our leopard seal recall device. And you can swim. One of the traditions at the station is when the ship leaves, it's a big deal. You're saying goodbye to friends that you've known forever, and they're going home. And as the boat pulls away from the dock, the tradition is to leap in to the water after the boat and sort of do a farewell wave from the water. And so people do this. All right, so let me talk about chemical ecology. So when you dive off of Palmer, you're actually diving in a forest. You're diving in a forest of macroalgae. And there are about 150 species of macroalgae, about a dozen that are really, really common. And one of the things that we've been interested in is, are these forests chemically defended? And why should they be? Unlike the kelp forests of California, you don't see sea urchins hanging on these things eating them. But one of my graduate students discovered that if you take a very fine mesh bag and you drop it over a macroalgal plant and you bring it into the lab, within that, 1 cubic meter of water that you've sampled, there may be 300 to 400,000 amphipods, little tiny crustaceans, herbivorous, living on these macroalgae. How are these macroalgae surviving? Well, we discovered that most of them, by and large, and particularly those that are large and dominant in their abundance, are chemically defended. The amphipods won't eat them. Other potential predators won't eat them. It kind of begs the question, what are those amphipods eating if they're not eating the macroalgae? And there's been a neat twist on this, and Chuck Amsler is really the one who's responsible for this neat study. But it turns out that there's algae that live in algae. They're called endophytes. And these little endophytes are very common in Antarctica, and they sprout from the algae, the large algae, and they send out a little filament. And it turns out that these amphipods think that this is candy. They eat the filaments, and that's where they get their food. Meanwhile, the macroalgae itself, the large one, is being kept clean. And one of our doctoral students brought the macroalgae into the lab and took away the amphipods and left it in the tank for a month, and it looked like it had a beard. It was completely covered with these filaments. So the amphipods are eating the little filaments of endophytes that are coming out of the macroalgae. It's very interesting. The other thing that dominates the seafloor in Antarctica, particularly when you go to McMurdo, are sponges. 55% of the seafloor is covered with sponges in Antarctica. Sponges are known to harbor chemical defenses. So this was one of our first target groups that we really looked at. We tested about 33 species of Antarctic sponges in feeding bioassays to see if they tasted good or not to potential predators. Well, I already told you that there's really not many fish in Antarctica that would eat these things. But you know what there are? Starfish. Starfish are incredibly important in the feeding ecology of Antarctica. You see them on the sponges, and Paul Dayton at Scripps did this great study back in the 60s and 70s showing that starfish are eating sponges like crazy here. So that was a good test organism for us. We use sea stars in our assays. Some of these sponges are 7 feet tall, and you can put a scuba diver inside the bays of the sponge, and they may live very long periods of time. They may be the redwood trees of the seafloor in Antarctica. And some of the sponges are covered with fouling organisms. This is a diatom, a sort of type of phytoplankton that's growing on the sponge. These can smother a sponge if they get too thick. And so we also found that some of the chemistry that we were finding in sponges could prevent fouling as anti-foulant compounds. And soft corals, another group that we've studied that have chemical defenses. Nudibranchs. My goodness, everybody knows that naked snails are filled with nasty chemistry. Well, if you have to be, you have no shell to hide in. So Antarctic's nudibranchs, every one we've looked at, filled with nasty chemistry. See, this one even says, I taste terrible. And then things like sea butterflies, little tiny pteropods that have lost their shells, moved up into the water, and evolved little wings. They're called sea angels. And there are so many of them sometimes that they're almost like stars in the sea. It's just phenomenal. You dive and you see thousands of these little orange sea butterflies, and they're all flapping their wings very quickly, even though it's freezing cold. Not surprisingly, they're chemically defended. But one day I was out at the ice hole and dive hole, and I looked down and I saw a little shrimp-like amphipod swimming along with a little orange backpack. And I said, what in the world is going on? So we put a fish biologist, John Jansen, and I got together as a collaborative team, and we sampled all around the sound. We put nets down. We caught hundreds and hundreds and hundreds of these little shrimp, and 70% of them were carrying a sea butterfly on their back. And if you took the sea butterfly off the back of the little shrimp, it would open its wings and fly away. There was never one that was dead. So why would this little shrimp go to all this trouble? Well, we did an experiment in the lab. We found a fish that's a natural predator, and if you offer that fish a piece of fish tissue, it'll eat it every time. If you offer the fish the little shrimp, it'll eat it every time. But then, guess what happens when you offer it the sea butterfly? The sea butterfly goes in the mouth, spit right back out. And if you offer it the shrimp with the sea butterfly in the mouth and then right back out. And the little sea butterfly swims away happily, carrying its abducted species. So this was the only case in nature that was known of 2 species where one abducts the other and carries it around. So it was published in Nature, which is a very prestigious journal because of its such a unique interrelationship. And driven in part, of course, by chemistry. These are Nemertean worms that we found have a neurotoxin in them. It's a protein. It's actually medicinal in the sense that there are neurotoxins in these worms that are used to look for pain kinds of mediators. And we named this particular compound. When you discover a new compound, you get to name it. It was named parbalazin after the genus Parbalazia, these large Nemertean worms. And then echinoderms themselves, starfish and sea urchins and crinoids. And these animals often have defensive chemistry. And we've looked at a lot of these species as well. Tunicates, another group that's found to be chemically defended. They do it in an unusual way, though. Many of them have a mucus that's very acidic. So they can have a pH of 2 in their mucus, and that's a good way to keep things from wanting to eat you as well. All right, so now let me kind of change gears a little bit and talk about drug discovery. Okay, so I am not a natural products chemist myself, but Bill Baker is. So I've learned a lot from him over the years about how you approach searching for new drugs in natural habitats. And that's what I want to talk a little bit about. And my university and NSF have been very happy that we're doing this. We do not get funded by NSF to do drug research. But when you write an NSF grant, there's always a section of the grant called broader impacts. And they're looking for relationships between your basic work and more applied work. And this has always been a wonderful thing, that we can say that we take all of the chemicals that we discover, all of the crude organic extracts that we take out of these organisms, and we can send them off to pharmaceutical companies, to the National Cancer Institute, to the Cystic Fibrosis Research Center at my university at UAB, and they can be screened to see if they have the potential to become a drug. So toxic chemicals can be drugs. All right, so chemical diversity. So you're probably familiar with these terms: pharmaceuticals, nutraceuticals, chemicals from foods, agrochemicals, antiphthalates, cosmetics. So these are all chemicals that have come from this drive to look at the discovery of new compounds. We've gotten into a world now where a lot of things are made in the laboratory. You know, combinatorial chemistry, where they'll find a particular chemical compound and they can modify it slightly chemically and then retest to see if they've increased the activity against a cancer cell or something like that. And you can produce chemistry in the lab. You can also go out and let millions and millions of years of evolution do it for you by looking at natural products. So I forget what, 60% of the things that are in your medicine cabinet come from plants, terrestrial plants, things like aspirin. So we look, our specialty is looking at what are called secondary metabolites. So they have no known role in primary metabolic processes, like glycolysis and these kinds of things. They tend to be small compounds. They tend to fall into different groups. They call them alkaloids and diterpenes and sesquiterpenes and on and on. And just to give you an example of things that you might know that are natural products, I just mentioned aspirin. It comes from willow bark. Penicillin from a mold. Are you familiar with Taxol? Have you heard of Taxol? What is Taxol used for? Breast cancer. It's a compound that's used to fight breast cancer. This was a real issue because it came from the bark of the yew tree. Well, they couldn't synthesize it in the lab. They're having a lot of trouble. Guess what happened to all the yew trees? Yeah, they were disappearing very quickly. And they finally figured out by taking a bacteria to produce part of the compound and putting it into the rest of the compound, they could synthesize it in the lab. They didn't have to use the yew tree anymore. These are other things that are secondary metabolites you're familiar with, things that smell: cinnamon, rose, vanilla, pine, etc. So about 60% of drugs are natural products, okay? 150,000 or more from terrestrial systems. Look how many we've found from the marine realm. A much smaller percentage, right? Very few marine natural products. This has nothing to do with the fact that there are fewer potential drugs in the sea. This is because the sea is a frontier. We haven't looked. So this is a whole wide open area. Why should we look in the ocean? Well, it's the seat of origin of life. I mean, things have been there a very long time. The chemistry has had a lot of time to evolve. There's lots of good reasons why the ocean is a wonderful place to look. So we've looked at these various different places. And where do you find new chemical diversity? It's in the oceans, it's in the polar environments, places where people really haven't looked. And you can see this here, that more than 95% of these natural products that have been found to date are in temperate and tropical latitudes. There's been very little done here in the poles. All right, so I'm not going to go into a lot of chemistry, but basically what we do when we do this is we collect the organisms, bring them in the lab. Thankfully, we do not have to collect huge amounts of organisms. We don't have to clear the seafloor of hundreds of square kilometers of something. We can freeze dry the tissue, and that's like making freeze-dried coffee. You use very low temperature and suction to essentially vacuum freeze dry. And then we put these dried tissues quantitatively into solvents so that we pull out these secondary metabolites. And we typically have a polar solvent and a non-polar solvent that we use. We take those and we test them first in our ecology assays, and then we send them off and they get screened. So let me tell you so far what some of the interesting things we've found that have the potential for drugs. This is a sponge from McMurdo Station called Lucetta leptorhaptus, and it turns out that it has a compound that has been named rapsamine that is active against nasopharyngeal cancer in bioassays. This is the green sponge, very common sponge in McMurdo Sound in the Ross Sea. It's about the size of a basketball, Latranculia apicalis. And as we found with a number of these sponges, the pigments that give it its color are bioactive. In this case, active against bacteria. They have antibacterial activity. And it's a group of, well, there's now probably 6 or 7 of these different dischlorhabdin compounds that have antibiotic activity. So here's the most exciting one. This is the one that potentially someday, I think, might become a drug. And even then, it's a long shot. Because as you probably know, producing an over-the-counter drug or something that's prescribed is very difficult and very expensive. But in front of our station at Palmer, the seafloor is often covered with these orange blobs. And they're about the size of baseballs. It's a colonial tunicate. Tunicates belong to the same group of the phylogenetic tree that you and I do, but they're a blob, okay? When they're larvae, they're called tadpole larvae, and they have the 4 basic vertebrate characteristics: pharyngeal gill slits, notochord, all that stuff you learned about in introductory biology. And then they metamorphose into a blob, and that's how they spend their life. Well, it turns out that the blobs have this compound in it that we named pomerolide. You get to name the compound. You're not allowed to name it after yourself, but you can name it. So we named it after the station, Palmer Station. So then we sent it off. So the National Cancer Foundation, National Cancer Institute, screened pomerolide against 25 different cell lines of cancer, everything from breast cancer to prostate cancer, you name it. And they called us up and said, we're excited about this because we found that this compound is very active against only one type of cancer, and it's very potent. It's a very, it has activity at very low concentration. So those are a couple things that work in your favor when you're looking for something. Has specificity. So it turned out to be melanoma, skin cancer. So the National Cancer Institute said, can we work on this? Would you be okay with us working on it? We have our own labs. And we said, sure. And so they do it free. And they took it. And one of the things you want to know about a potential drug is, can you deliver it to a tissue? So they did a drug delivery test. So how do you do that? Well, you take a little tube, plastic tube, and you put some cancer cells in it or what have you. You then essentially take the compound and you label it, radio label it, and you inject it into a mouse. You've taken that little tube and you've put it under the skin of a mouse, and then you wait to see if the radio label shows up in the tube eventually. So it can, the compound can move through the tissues. And it passed that test. Then another test is, can we make this in the lab? Because tell you what, it's very expensive to take a bunch of ships down to Antarctica and harvest this. It ain't going to happen, right? So you have to be able to produce the potential drug in the lab, benchtop, economically, right? And it passed that test. Then you have to know how it works. Okay, is this a new mode of action? Because if it's a replicate of what something else does, it's probably not as attractive. So this is a sodium ATPase inhibitor, which means that you sort of starve cells that are multiplying of the energy that they need, ATP, and they die. So there is one other compound that has sort of a similar, slightly different mode of action. So unfortunately, we're not the only compound like that. But so the NCI worked on it for a while, and then some pharmaceutical companies got interested, and so that's kind of, here's the data for the activity against melanoma cells. It just shows essentially that it kills them at very low concentration. And it's those low lines that are where the activity is reflected. So how do you possibly get something to market when it comes from a natural product? So this is really amazing to me, not being in business, but it takes years, you have to go through all these tests to make sure you can make it in the lab, purify it, test it in some animals. Then you have to go into human clinical trials, which it has not done yet. And look how long that can take, 1 to 4 years, 2 to 10 years, 5 years average. And then you have to apply for applications. So it's a 10 to 15 year process to get a drug to market. It's not easy to do. And it's extremely expensive. $1 billion, I mean, hundreds of millions of dollars to get something to market. So you have to find investors, you know, that are willing to do it. This is another compound that we've found that's in Antarctic macroalgae. It's a protein that's very active against the H1N1 flu virus. And we did some assays. This is a control where we're looking at activity, and you essentially see that the cells here, this viral cells, are essentially not affected when they're treated with this particular protein. All right, so that's just a summary of this particular compound. All right, so I'll just finish up and I'll take questions from you. So we've kind of, our group and maybe 2 or 3 others have sort of debunked this idea that Antarctica would not be very interesting from a chemical perspective. The biodiversity is very high there. It's an extremely ancient system in a geological sense. So there's been a lot of time for evolution between predator and prey. Even though there's no fish, there are a ton of predators in the form of starfish. So there's lots of reasons to have chemistry. So it's really been a neat place to work in that sense. And of course, now this entire system is being threatened because the changes that are happening, at least along the peninsula, are very, very rapid. Warming, ocean acidification. And so when I talk about climate change, I often point out, because people are like, who cares? Antarctica. But if you can convince them that there might be a cure to a human disease, you might get their ear, or Congress's ear. So that's important. All right, well, thank you, and I will open the floor to questions. Okay. All right, who wants to go to Antarctica? I'm now leading trips to Antarctica, believe it or not. My climate change theme has gotten me invited to lead a cruise to Antarctica every year by a cruise company. So if you have a rich uncle and you want to go to Antarctica, you can go with me. I'd love to show it to you. I've taken 6 or 7 of my undergraduate students from my university to Antarctica, and it's just an amazing experience. And it's neat because the cruise is philanthropic, and they allow me to purchase a piece of equipment that we give to the scientists at my station when we visit for a day that's related to their climate research. And the 200 guests on board are like, this is the best thing I've ever done. You know, they just love it. So it's a really neat way to educate people about Antarctica and turn them into ambassadors. Yeah. Yes? What's the next step in my research? So right now, I'm finishing up, I have 3 NSF grants. One of them is finishing up a study of the effects of ocean acidification on some Antarctic species, mainly shelled invertebrates. So we're still in the process of publishing some papers. The chemical ecology program, I think, is probably the answer to your question. That's the next step, because we were just refunded for another period of 4 years, which means that we will have 3 trips to Antarctica. And then very wisely, the NSF says, no, you can't go back to Antarctica in the last year of your funding. You have to stay home and write, because they want you to produce the papers, okay? There's lots of scientists that like to do the research, but they're not as good at getting it published, right? So that's a really smart thing. So that's what we're going to be doing. We're going to continue to look at the interactions between the macroalgae and their predators in terms of the chemical ecology, because that's such an important ecosystem along the coast. The other project that I mentioned is looking at king crabs, and we've had that refunded twice. It was funded originally on what they call a high-risk grant. I thought that was pretty cool. You get a high-risk grant funded. It means that there's a really good chance it may not yield much of anything. But if it does, it's really, really cool. And so they gave us the money, and we found the crabs. And so that's exciting. And they gave us another round of funding to follow the crabs. And we're looking at the interaction between the crabs and their potential prey and how they are potentially rewriting that benthic ecosystem that has never had a crushing predator in it now for millions of years. So that is another area that will probably continue. And then in terms of my other activities, writing. I've really gotten interested in writing books for the general public. So I haven't thought about the third book yet, but I probably will, yeah. You know, we have tried over the years a little bit of that. We went and bought one of these big refrigerated tanks you see at the grocery store that they put the lobsters in. And we bought 3 of those. We brought things back with us, which is not easy, you know, in coolers and shipping and all, you know, because it takes 3 days to get to Antarctica. Even the quickest way, you could get there by flying. So the short answer is that we've tried, but we haven't had much success. We get them back and the starfish sort of sit there and they don't feed and eventually they die. Now that said, there's a group in New Zealand that has been very successful at establishing the appropriate conditions. They have, unlike us, they have access to fresh seawater. They're right on the coast. The water's very cold and they can chill it further. And they are raising Antarctic starfish and sea urchins for work that they're doing in ocean acidification and some other areas. This is a laboratory in Dunedin that I did a sabbatical at, you know. Right, yeah. I have brought back invertebrates for the museum near the airport in Christchurch, New Zealand. They have a big tank. And the frequency at which they ask me to replenish the tank suggests to me that they don't do real well. So it's not trivial to keep them happy out of Antarctica. So you have to have a good reason to go to Antarctica to do the research. One of the quickest way to get your proposal rejected is to propose to do something that's really, really, really neat, but you don't have to go to Antarctica to do it. You know, you say, well, I'm going to go there and collect them and bring them back and I'll do this and they'll be fine. And everybody knows that that's probably not going to work. So other questions? Yeah. In general, yeah, I'm sorry. The question was, is there a latitudinal gradient in chemical defenses in benthic invertebrates? And the answer is, from what our work shows, our levels of chemical defense are very similar to what you find in temperate realms, but not quite as high as what you find on a tropical coral reef. So there does appear to be still some tendency towards more chemical defenses on tropical coral reefs. And if you've ever been out on a coral reef, you know that there's fish everywhere. The level of predation is very intense. So I think that might explain it. But in the true sense of a latitudinal gradient, no, because our levels are just as high, in some cases higher than those that have been found in temperate species. And we've looked at, particularly in sponges, found this. Yeah. Ah, okay. So what is the motivation for me to write popular books? Well, I am now a senior professor. My university gave me an endowment, and I don't have to worry about getting tenure. I don't have to, you know, so I have the freedom to do some things at my point. I would never have written popular books as a beginning assistant professor. It would have been academic suicide. But now I can do it, and I enjoy it, and I think it's really important. And the NSF really likes it because they like to see the scientists that they've supported over the years give back. And one of the ways you give back is you share what you've found through their funding with the general public. That's a broader impact. So I have a lot of support from the federal agencies. My deans love it. President likes it. They can show me off to the, you know, whatever. I'm one of the faces of the campus now, so, and I don't get paid for it. It's terrible. I now have 2 students that are my, what do you call the social media assistants, and they are tweeting. In fact, they told me, would you take a picture of me? They said, they said that I have to get pictures all day today because they are tweeting this event and you guys. So a couple of pictures, maybe the room too, yeah. So they got a Facebook, they got me, I've got a Twitter site, and my books, or whenever there's something written about my books, like my book that just came out was featured in Science News and Scientific American, they take that and they tweet it. So anyway, to get back to your question, the answer is that I thought to myself, how do I write? Well, I'm trained in science writing. I'm not trained in writing for a popular audience. But when I was in high school, my favorite class was English. I started out college as an English major before I discovered marine biology, so I knew I had some interest. And they said, well, I'm going to do this. So what do you need to write a book? And I asked around, and you need a literary agent. Well, why do you need a literary agent? Because the publishers won't look at your proposal unless you have a literary agent. This is called a catch-22, okay? So I thought, well, how can I use my connections to make this happen? So I just so happened to be a very good friend of E.O. Wilson. And this is because when I was a young professor, I invited E.O. Wilson to come to UAB to win this big award, and he won it. But nobody told me when I nominated him that I had to host his visit for the whole week. So I had to pick him up in the morning and have lunch with him and take him home at night. And we had these long discussions and we kicked ant mounds together. And the next thing you know, we're friends. And he has taken kind of an interest in me. And so when I thought, well, who writes books? Well, E.O. Wilson writes books. And he writes pretty good books. And so I told him I was interested in writing, and the next thing you know, he called up his literary firm here in Boston, and they took me on as sort of a chance, right? So now I have a literary agent in Boston, and they helped me craft a proposal of Lost Antarctica, and they convinced me I had the whole book in my head. I didn't need to do any library research because I've been working there for 30 years, and what people want to hear is stories. And so we got a company, a publisher to take it on. And now I've discovered once you have a book, it's much easier to get your second book because it was successful. You know, it met its advance, even though it was a small advance. But anyway, the same publisher said, we're ready, you know, where's your next book? So once you get in the door, so that's kind of how I got into it. And then I discovered I can write. I can write because every morning for 2 hours, I sit in my office and I write. Even on the weekends, I'll write. We'll be driving off to the coast for the weekend. I'll be sitting there. My wife will be driving. And for 2 hours, I'll get 250 to 500 words down. That's not that hard. And at the end of a month, you have a chapter. And at the end of 8 months, you have a book. So some of writing books is time management. You have to kind of squirrel away a little time. And if you're the kind of person that says, I'm going to put it off till Friday, and then the next Friday you say, I'm going to put it off to the next Friday, it must be very hard to write a book. Those are the 10-year books. So that's how I got into it, yeah. Good question. So when I said there's no fish, it's a little bit of a, I'm sort of making a point. And the point is there are no fish in Antarctica that are capable of driving chemical defense because they don't have strong crushing jaws. They're not durophagous fish, as they're called. So this means all the fish in Antarctica are wimps. They eat little tiny crustaceans, you know. You can swim up to a fish and pick it up and look at it and put it back down. These are not burst swimming pelagic fish, okay. Now, there are 250 species of fish that have, it's a wonderful example of what we call adaptive radiation, because what happened is when Antarctica separated from South America about 40 million years ago, and the Antarctic circumpolar was established and locked Antarctica into a deep freeze, only one family of fish, the Notothinidae, was capable of surviving at low temperature. And the way they did it is they evolved an antifreeze. So just like you put in the radiator of your car, this is a compound that prevents ice crystals from forming. It actually is a glycoprotein that prevents ice crystals from touching one another physically. So if you look at the blood of an Antarctic fish at McMurdo where the water's sub-freezing, there's tiny little ice crystals in its blood, but they're not touching one another. They're not allowed by that glycoprotein to form a solid block of ice that would kill it, and they'd flash freeze the whole animal. So that's kind of an interesting story. The guy who discovered this, Art de Vries, I know Art very well because he's gone to Antarctica 50 years in a row, and he's an old cudgmudgeon now. But Art is the father of the glycoprotein. He discovered it in 1957, published it in Science, and all sorts of neat work has come out of that. You know, they've now know the genes, and they're trying to figure out how to take this antifreeze and stick it into a tomato, so that you don't have to have cardboard tomatoes in the winter. You know, it would be possible to keep tomatoes from freezing and growing, and they could grow in colder places. So there's all sorts of sort of applied angles on this antifreeze. So yes, there are fish, but they're not really in the chemical ecology realm. Yeah. Because if you have a mouth that's capable of crushing something, you potentially could take a big chunk out of a sponge. You know, sponges have skeletal elements called spicules in their tissues, so they're not real soft and fleshy. You have to have some jaw strength to do that. So I just believe that, and you never see them grazing any of these things. So I think that you just don't have grazing fish that are feeding on those kinds of things that would produce chemistry. Some shelled invertebrates have defensive chemistry. Brachiopods, we found a brachiopod looks like a clam, but it has a little stalk. What's it called? A peduncle? The little peduncle stalk, and we found that the peduncle was chemically defended. The tissues inside the shell were not. So there are some shelled species that take advantage of chemical defenses. But yeah, you just don't see anything. Have you snorkeled on a Caribbean reef? Have you ever listened? Have you ever heard the crunch, 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 crunch sound? You know what that is? That's parrotfish eating hard coral. You know, there's nothing like that in Antarctica. So you really don't have that selective agent for the fish driving things. But what you do have are the sea stars, much more than in temperate and tropical systems. They're very important in the ecology. Yeah. You have to wait for tonight. Yeah.
I was just hoping that you could repeat to the people online and everybody here, your talk is at 5 o'clock in Barnum 8, the title of your talk, and we look forward to seeing you there.
Okay. Well, my talk tonight is in Barnum 8. It will be at 5 o'clock, and the title of my talk will be from penguins to plankton, studies of the marine ecology of climate change in Antarctica, something like that. Thank you.
Professor McClintock's books will be available after his talk. People would like to get a signed copy. And next week, stay tuned. Solar Power Comes of Age, Philip Warburg. Same place, same location, same time next week. Thank you so much again.